Good morning, friends. So, in this lecture, today I am going to discuss you about the history of animal genetics. So, actually, the history of genetics in general we will discuss here, but there is a separate history of animal genetics that we will be discussing in some other class. So, today I will be concentrating more here on the brief history of common genetics. So let us start word genetics with word genetics. What is genetics? So genetics, we know that it is a science that deals with the heredity and variation. So many a times we are getting confused that genetics is the science which only deals with the heredity, but it is not like that. Genetics is the science which deals with the heredity as well as variation. So it tells that how things are getting transmitted from one generation to another generation, and during that transmission how this variation is going to arise or how this variation is arising. So this is what genetics is. Then the question comes, why we want to study genetics? So, the study of genetics is generally, we are studying genetics or we are studying any science to solve the myth as well as the various wrong assumptions that has been encrypted in our life which need to be solved scientifically. So, with this aim, the genetics was started. Now, coming to the history, so we know that understanding of origin is a constant pursuit of a man. So man always desire to know how the man has been originated, how it has been evolved, how human is evolved. So what it was earlier and how it is evolved. So in this series, to explore this pursuit, the effort started in 1858. Understanding of origin of species and how species variability arose was revolutionized with the work of Darwin and Wallace. So we are all knowing about the Darwin, and one more scientist was there, Wallace. So this is Darwin and this is the Wallace. So with this understanding of origin of species and how the species variability arose, it was revolutionized with the work of Darwin and Wallace, and that led to the theory of evolution via natural selection. So on the left hand side, I have shown the photographs of scientists. You just get an idea that how those scientists were looking like. In 1859, the book Origin of Species was written by Charles Darwin. Then coming to the 1866. The 1866 was the breakthrough year when Gregor John Mendel, he gave the concept of heredity and how the characters are getting transmitted from one generation to another generation. He just conceptualized the idea and he come to a conclusion that the inheritance is particulate in nature. That means there are certain particles. Actually, at that time, the gene was not identified or the term gene was not coined. People were not aware about the gene. So he told that there is certain particles who are responsible for transmission of character from one generation to another generation. And he was rightly said as the father of genetics. Then in 1866 itself, Haeckel states that hereditary material reside in the nucleus. So the thing that Mendel told that okay, there is certain hereditary material, those materials are particular in nature. But in 1866, Haeckel first told that these hereditary materials resides in nucleus. And in 1971, Misser termed them nuclein. So this is Haeckel. Then in during the year 1879 to 1892, the scientists Fleming, Strasburger, and Benden. These were the scientists who worked and come with the technique of accurate counting of chromosomes. They coined the term chromatin, mitosis, cytoplasm, nucleoplasm, prophase, metaphase, etc. So there was certain development during this year 1879 to 1892, and these scientists worked and found they gave certain terms which now we know, but previously it was not known. Then in 1892, August Weismann, you might have known his name, you might have heard in your plus 2 classes. The August Weismann, he told that heritable information is transmitted only by germ cell in the gonad, but not by the somatic cell. And during this work, he gave the germplasm theory of inheritance, theory of inheritance of germplasm. So the August Weismann, he told that the inheritable material are getting transmitted by the germ cell. Then in 1889, Francis Galton, he told that he studied the variation by quantitative measures. He studied the variation by the quantitative measures of difference among the traits. So he actually gave the concept of biometry. This study, when he quantitatively measures the variation among the traits, and this study gave rise to the science that is called as biometry. Bio means biology, metry means measurement. So measuring the biological things. So actually he mixed the mathematics with the biology. In 1894, William Batson, he gave the concept of discontinuous variation. Initially it was thought that the variations are getting transmitted continuously, but he was the person who told that no, this variation is not continuous but it is discontinuous, and he worked on certain plants and he come to this conclusion. In regarding his work we will study in the coming classes. Actually the study of history means that you are covering whole of the subject, because we have to actually in this history all the terms that we are going to study in this unit 2 or everything will come, but we will study those terms in the latter part of the course. In 1894, Carl Pearson, what he did, he gave the methods for analysis of statistical frequency distribution. So he applied statistical frequency distribution in the biological science, and actually he gave the statistical analysis of the biological experiments or results, or whatever the results of inheritance experiment he is getting, he analyzed it biologically with the help of statistical frequency distribution. So he is Carl Pearson, he is William Batson, and this is Francis Galton. Then in 1900, Karl Correns, Hugo de Vries, and Tessermark. These three scientists independently rediscovered the Mendel's work. So initially, if you remember, the Mendel has given his concept of inheritance in year 1866, but the work at that time, it was not understood by many of the people, or it was not recognized at that time. But again in 1900, these three scientists independently, they worked and come to the conclusion that okay, what Mendel had said in 1866 is actually the true, and this is the actual concept of inheritance. So the Mendel's work was rediscovered independently in 1900 by these three scientists. This is Hugo de Vries, then Karl Correns, and Tessermark. So these three scientists rediscovered the Mendel's law. Now, in 1902, Sutton and Boveri. Sutton and Boveri worked together and they told that chromosomes are a physical location where unit of heredity resides. That means they told that okay, unit of heredity, for which the Mendel told that the particles or factor, the Sutton and Boveri, they told that the particulate material or factor or genes which we know now, they are located on the chromosome, and that is why they gave the chromosomal theory of inheritance. Chromosomal theory of inheritance. Again in 1902, William Batson, he termed the coined genetics, homozygote, heterozygote, epistasis, F1, F2 generation, and allelomaps. So what are these terms that we will study in the latter part of this course, but you just try to understand that this William Batson was the man who gave these terms. Then in 1903, Wilhelm Johansen in 1903, he gave the concept of phenotype, genotype and selection. Again in 1905, Batson and Punnett. So this is Batson and Punnett. So Batson and Punnett, they gave the concept of linkage. So regarding linkage also, we will study in the latter part of the course. When this course advances, we will study about the linkage, and there we will again study how Batson and Punnett discovered the concept of linkage. Now in 1908, Hardy and Winberg, these two scientists, they gave the principle of genetic equilibrium. Actually, Mendel gave the concept of inheritance with the traits which are controlled by either one or two or few genes. But today we know that most of our traits, most of our economic traits in animals, they are controlled by polygenes. That means more than one gene or more than few genes, when many genes are controlling a particular trait. So how the Mendel's principle is getting extended to this polygenic inheritance, that was the rule for that was given by Hardy-Weinberg. So that is why this Hardy-Weinberg law is considered to be the extension of Mendel's law. Then in 1910, Thomas Hunt Morgan, T.H. Morgan demonstrated the sex linkage in Drosophila. So he was the person who gave the concept of sex linkage in case of Drosophila, and he gave the concept of linkage group. So what is linkage group? In 1919, he gave the concept of linkage group. So what is linkage group? The linkage group is actually the haploid number of chromosomes of any individual or any species. So he was the person who gave the concept of sex linkage in Drosophila. In 1903, Startwent first gave the first genetic map was developed by Startwent in Drosophila. So what is genetic map? Actually, we know that now in on chromosomes, the genes are located at different places. So what are those places? What are the neighbors of that particular gene? So above that gene, below that gene, so all the maps, how the genes are located on chromosome, the study of that structure is called as genetic map. So Startwent in 1903 first gave the genetic map in Drosophila. So how genetic map is developed, that also we will study in the when the course advances. Then in 1914, C.B. Bridges, he observed the observation of non-disjunction in sex chromosome. So he observed the phenomena of non-disjunction. So what is non-disjunction? Again, we will study in the later courses. Just you remember that C.B. Bridges was the scientist who gave the concept of non-disjunction. So actually this is Hardy, this is Weinberg, then this is T.H. Morgan, this is H.J. Muller, then this is T.H. Morgan, and this is C.B. Bridges. Okay, C.B. Bridges. Now, in 1927, H.J. Muller, you might have heard his name because he was very famous for his work on X-ray, and he demonstrated that X-ray can induce the mutation. He told that X-ray can induce the mutation, and he demonstrated how X-ray is inducing mutation. And again in 1948, he gave the concept of dosage compensation. Dosage compensation. So what is dosage compensation? We will study in the as we will proceed into the unit 2 further. But right now you just remember that dosage compensation theory was given by H.J. Muller. Now in 1928, Frederick Griffith, he gave the concept of transformation. So what is transformation and why this is important? Because transformation was the first experiment which was helpful in proving that DNA is genetic material. So you might have studied in your microbiology classes about the transformation. If you as we will proceed in further into the lecture, we will also get some idea about the transformation. So Frederick Griffith was the person who gave the concept of transformation. In 1941, Beadle and Tatum were the two scientists who gave the concept of one gene, one enzyme hypothesis. One gene, one enzyme hypothesis. They gave the concept that one gene is synthesizing one enzyme. But now the concept has changed. Now it is one polypeptide, one enzyme concept. So that we will see in the later classes. So the one gene, one enzyme concept was given by Beadle and Tatum. Then in 1941, Mather, he coined the term polygenes. Polygenes already we are knowing, or we will see in the later classes what is polygene, what is pleiotropy, what is polygenic inheritance. So these all we will study. In 1944, again Avery, MacLeod and McCarthy, these three scientists proved that in experiment they proved that DNA is the genetic material. DNA is the genetic material. Then in 1946, Lederberg and Tatum, they demonstrated the bacterial genetic recombination. How bacterial genetic recombination takes place, because this was helpful in study of the variation, or how the hereditary materials are getting changed when it is getting transmitted from one generation to another generation. Then in 1950, Irving Chargaff, he gave the concept of base pairing law. We know that our genetic material is composed of 4 nucleotide bases A, T, G, C. So, and we also know that A is getting paired with T by double bond. Then G is getting paired by C by triple bond. So how this A is getting paired, G is getting paired with C, this law actually it was deciphered by Irving Chargaff. That is why it is also called as Chargaff's law. Then in 1952, Hershey and Chase in their blender experiment, this is Hershey, Chase. Okay, Hershey and Chase with their blender experiment. In blender, they mixed the DNA content. We will study that particular experiment, and they confirmed that okay, the DNA is actually genetic material and not protein. There was confusion that protein may also be genetic material, but they proved that the DNA is actually genetic material. Now, in 1953, Watson and Crick, these two scientists deciphered the structure model of DNA. The structure of DNA actually it was revealed by Watson and Crick in 1953. And in 1957, these two again they gave the concept of central dogma of molecular biology. So what is central dogma of molecular biology? We will study later. As of now, or you might be knowing, but as of now you just remember that central dogma of molecular biology was given by Watson and Crick. Then in 1955, Okawa discovered the RNA polymerase. In 1956, Korenberg discovered DNA polymerase 1. In 1958, Messelson and Stoll, they gave or they discovered that DNA replication is semi-conservative. Suppose this is the DNA and the strand, the double stranded DNA, then strand name is A and B. So now after replication, what will happen? The two molecule of DNA will be there, that means 4 strand. So this A will be the older one and this C will be the new synthesized one. Here B will be older one. Now, okay, again we will see. This B is the older one, whereas D will be the synthesized one. So, by this he proved that DNA replication is actually a DNA replication is semi-conservative. DNA replication is actually a semi-conservative model. Now, Jacob and Monod in 1961, he gave the concept, he discovered the lac operant model of gene expression and regulation. How genes are getting expressed. That you might have studied in your +2 classes, how genes are getting expressed, or you might have studied in biochemistry classes also, or you will study in the biochemistry classes. In 1961, Nirenberg, he told that each amino acid corresponds to a triplet codon. That means suppose there is methionine, an amino acid is methionine. So methionine is getting, or methionine is denoted by a triplet code that is AAU. So this is the triplet code correspond to amino acid. So likewise, all the amino acids are getting signified by a triplet codon. So this was first deciphered by Nirenberg. Now, in 1970, Arbour and Smith, these two, they gave the first restriction enzyme. You might have studied in your microbiology classes. Restriction enzyme. What restriction enzyme can do? They can cut the DNA molecule at a specific site. So this was the very important discovery when we want to manipulate the DNA. We want to instill a desirable change in the DNA. So that discovery was considered to be very important, and they were awarded with the Nobel Prize also. The Arbour and Smith, the first restriction enzyme they discovered. Then in 1970 itself, Baltimore and Temin, they discovered the enzyme reverse transcriptase. Reverse transcriptase or transcriptase. From where? Because we know that DNA will form RNA, and RNA from RNA we will get protein. But what if this RNA is synthesizing DNA? That will be synthesized with the help of reverse transcriptase enzyme. Okay, so this was first discovered by Baltimore and Temin in 1970. Now, Paul Berg in 1972, the first recombinant DNA molecule, it was made by Paul Berg in 1972. In 1975, Irving Southern, he discovered the technique of Southern blotting or Southern hybridization. So you will study in your microbiology what is Southern hybridization, Southern blotting. In 1977, Sanger and Gilbert, they developed the method of DNA sequencing with the help of chain termination and chemical method. So DNA sequencing means how the bases, nucleotide bases, that is A, T, G, C, how these bases are arranged on the DNA. So that is deciphered with the help of DNA sequencing, and it was first discovered by Sanger and Gilbert by chain termination and chemical method. Now, in 1973, actually this was the discovery which revolutionized the molecular biology and genetics. So in 1973, Carey Mullis, he gave the procedure to amplify large amount of DNA, and that procedure is PCR, that is polymerase chain reaction. Polymerase chain reaction. So this polymerase chain reaction, what it did, it is a technique of in vitro synthesis of DNA with the existing DNA. So this has revolutionized the whole of the molecular biology, and for this he was awarded with the Nobel Prize also. In 1990, Human Genome Project was launched. Human Genome Project means the sequencing project, whole genome sequencing project of the human. So this was also a very important period. In 1997, Ian Wilmot, he first cloned the sheep. With somatic cell. Somatic cell of a sheep was taken, and that was developed into a in a sheep, newborn sheep. So Ian Wilmot, he first cloned the sheep, and the name of that sheep was Dolly. And in this series, later on in India also, in buffalo, NDRI developed the cloned buffalo with this method or the modification of this method. So this was about the general history of genetics. So I tried to summarize the year-wise development as the genetics progresses, but I have missed many things, and this maybe means there has been lot of development during these 100 or 150 or 200 years. So, but the summarizing everything was very difficult in this small lecture. So, as we will advance in the course, we will study then and there how the things were advanced. So, thank you very much for your patience listening. If you have any confusion, you can ask me either in class or on the social media platform where I will be uploading this lecture. Thank you. Thank you very much.